Good afternoon, everyone. Thank you for the kind invitation. I'm very happy to be here and present what we do at BioExcel. Hopefully, the applications that I'm going to present will be useful in your research. I know quite a lot of researchers and also industry in Greece is using some of these codes. So, if there are ways to help you with their usage, we'll be very happy to do that. As Lena said, I work at the KTH Institute of Technology at the Supercomputing Center there, PDC. As Lena mentioned, Europe has been investing very heavily into the deployment of large-scale supercomputing resources across Europe, and many of the countries already have different size systems with continuous deployment of new machines. But efficient utilization of these machines is not very trivial. They give a lot of power and compute capability, but still to make best use, one needs some expert knowledge of how to do them. Which is why, in parallel to the hardware investment, Europe invested in the funding of centers of excellence, alongside to the initiative on the national competence centers such as EuroCC Greece. And while the national competence centers are focusing on supporting the local communities within the country, the centers of excellence are organized per domain, and they cover very different fields. And today we will hear from me for BioExcel and also from Andrea about CombBiomed, one of our partner centers of excellence in the life science domain. So life science research spans a very wide range of scales, from atoms all the way up to individuals and population simulations. It's impossible to cover in one initiative all of that, which is why different initiatives are working heavily on different aspects of that. And in the case of BioExcel, we are focusing on the lower scale, on the molecular systems up to cellular level. Later, Andrea will talk more about how HPC can be used for simulations of higher level structures, tissues, organs, and so forth. In the case of so for life sciences, some of the needs for HPC come from the fact that now we have at our hands we either have solved or we can easily model the structure of all most of the proteins that build our bodies, and there's tens of thousands of those, and understanding their dynamics and structure allows us to for example, to design faster and more effective drugs. And HPC is a very powerful tool to, for example, screen through hundreds, thousands of potential drug compounds and how they can affect protein structure. Which is why BioExcel was established to provide life science researchers with, need to emphasize here, high quality and user-friendly software. Because all of this usage of high performance computing resources is, as I said, is not very trivial. So the quality and the usability of the software is paramount for increasing the productivity of researchers, which is another of the goal of BioExcel, to increase the expertise and the skills of researchers, and also, more importantly, to strengthen the community in the field. We've been running since 2015, and we're now in our third phase of funding until 2026. Hopefully, we'll continue longer term. And we work with the core developers of several widely used applications in the field, which I will present now. So one of the main areas where we're working extensively is the development of software for molecular dynamic simulations, GROMACS, which is one of the most popular codes in the field. I know many researchers who are using it in their work, and one of its main applications is for the simulations of biomolecular systems, proteins, membranes, DNA, other types of biomolecular structures, such as, for example, cellulose or others. Now, with the latest development of the code, one will be able to simulate very, very large systems on the order of almost half a billion particles. It is also widely used application for soft matter physics, like materials research. Some of the specifics of GROMOS is that it's been in development for almost 30 years now, and it's one of the most widely used applications. Some of its strength comes from the fact that it's very highly tuned to make extremely good use of all the modern hardware architectures, and through co-design work, working closely with hardware vendors such as NVIDIA, AMD, Intel, and others, the software has been very well optimized to make the best out of the CPU power that we have access to. And a lot of work has been put into the portability of the code, into the design of very advanced algorithms to run this simulation set, because a typical simulation includes billions of steps of integrating the equations of motions of atoms. Now, modern supercomputers are increasingly being built as so-called hybrid architectures, where we have not just standard CPUs, but also accelerators, GPU cards, and there's some other more exotic accelerator types, but the typical one is, if we look at LUMI, which is currently the fastest supercomputer in Europe, as shown here on the right, there's a very big partition, which is a machine which has CPU part and GPU with 8 GPUs on each of the nodes. Now, each of these nodes that you see here is a very powerful hardware, and LUMI has a lot of those. And to make best use of that, it's not very trivial. This is a bit of a technical slide, but just to give you an idea of what is needed by modern applications to make best use of such hardware. For example, in a typical case, when you have a serial code which runs just on the CPU and sequentially is calculating different type of interactions, bonded interactions, non-bonded, etc. When one has access to these powerful hybrid supercomputers, then you need to think very carefully how you can offload all of that to be done in parallel, both simultaneously on the CPU and on the GPU side. I will not go into details, but just this is to get an idea that it's not very easy to reorganize code, and the applications that I'm showing here are implementing already that. GROMOS is a big effort, mostly developed in Stockholm, the group of Erik Lindell. There's many contributors from around the world. One of the important, from pharmaceutical point of view, application of GROMOS is that you can use it for free energy calculations, which is already done by GROMOS, but to do it at scale and in an automated way, we've developed a tool called PMX, which automates the process of how chemical transformations, or one is able to mutate, say, amino acid or could be a nucleotide, and then calculate how this mutation affects the stability of the structure using free energy calculations. And also, what one can do is to calculate the relative stability of ligand binding. So in that way, you can screen through library of drugs and see which one creates the most stable structure. And we're showing how one can, for example, calculate, say, work with about a dozen protein ligand systems and go through almost 500 different types of mutations and synergic perturbations to screen through this library in a matter of days, which would typically take months in the classical way. If you don't have access to HPC resources, but you have access to cloud, for example, such as the one provided by Amazon Web Services, or you have local cloud clusters, it's possible to use this ligand screening in a high throughput manner, also very efficiently. And importantly, as shown here, these plots on the right, with GROMACS and PMX, one gets precision accuracy on par with very expensive commercial software. And PMX is developed by the group of Verdegroot and many other collaborators in Göttingen, in the Max Planck Institute. A third application I'd like to present is called HADOC, which is used for integrative modeling. HADOC is also one of the most widely used tools for docking studies. In docking, just very briefly, one is looking at calculating the stability of macromolecular complexes. So you can dock proteins, or you can dock ligands to protein surface or DNA or any other type of molecule. In a typical manner is like if you do not have any other external information, one is running confirmation sampling of the landscape, conformational landscape of these complexes, and one needs to calculate the interaction energy for these complexes, which can take different forms. Those conformational landscapes tend to be extremely broad, and it's very time-consuming to, sometimes even impossible, to explore all different states. It's very beneficial if one can limit the search, and one way to do that is to use additional information, so-called information-driven search, that limits the exploration of this conformational landscape. And what kind of information could we use? Well, it could be from experimental studies. It could be cryo-EM, SAGS, could be NMR or electron microscopy, as I said, or could be also included from different additional simulation studies. So you can integrate data from different types of simulations or screening, or if you have some bioinformatics data, for example. This HADOC offers this very broad integrative modeling platform with very broad capabilities for docking, and it's been consistently performing very well on different competitions, blind screening. For example, a few years ago, it's just one example where this shape restraint protocol that was part of the GPR-doc experiment. It's called one of the best performers. So if docking could be applicable to your research work, we would highly recommend to have a look at HADOC. And it's possible to use it in a high throughput manner, but also on HPC. We've done different studies and benchmarks. One is able to use thousands of cores in a workflow manner that finishes in less than a day. And the HADOC server is very popular around the world. There's like hundreds of countries, researchers from hundreds of countries are using it, with tens of thousands of jobs being run all the time. The main developer is the group of Alexander Bonvin at Utrecht University. So the last one I will introduce is the BioExcel building blocks, which is an automation workflow. Now, a typical research use case involves the usage of many different applications and tools, or maybe you would need to involve the same application with different parameters in the part of this of a given use case, and it can be quite complicated. Then not to mention that often one is working with a very big library of different tools. Now, even if you have access to HPC and the most time-consuming part of your work can be sped up considerably on the supercomputer, to enable to help with the productivity, what's very beneficial is if you can wrap all of these applications into a single workflow that can be deployed and run at once on the super machine, which is why what BioExcel building blocks was developed. It's just one. It's a workflow solution. There are other workflow solutions, but this is working very well in our case, where we developed different modules. So for every application, we have a different module which can be put together, linked to build a standalone workflow solution. Excuse me. Okay. Now, as of now, there are 17 modules that they provide functionality for the most commonly used, most commonly needed use cases, for free energy calculations, for even for quantum mechanical calculations, for docking, for MD with GROMACS, or if you're using other MD codes such as Amber, we have modules for that. Also for machine learning, virtual screening, and so forth. The library is continuously being updated. The new modules are being added. There's also a template for users to develop their own modules if you are interested in extending the capabilities. So the way BioBB works is that once you have a description of your workflow that you want to execute, one configures the BioBB. It's Python based, very straightforward and easy to configure. Then using a workflow management system, in our case we've been using PyComps developed at the Sloan Supercomputing Center. This workflow manager then distributes the job over all the nodes on the supercomputer. And we've done several different types of benchmarking. We can show, for example, that with an example system, the red line is the ideal scaling, the blue one is the achieved scaling that we have. This is called strong scaling, where we have the same system, we're just increasing the number of nodes, and each node has about 10,000, it has about 1200 cores, which gives this 500 is about 60,000 cores total. So we see that at around 300 nodes, we still have very considerable speed before getting to a saturation. Similarly, there are we can test the weak scaling, where with increasing the number of nodes, we increase also the size of the system, and there also you can see that at around 300 nodes, we are still doing really well with the scalability. What is also possible to do is to automate the process even further, that when you allocate resources to the workflow as needed, because in typical case, at the start of a workflow, you may have a few steps where there is no need for many resources, because you're doing some pre-processing configuration work. Here we see that there's no need for many resources, and but once you get to the core part of the calculations, then one can automatically, through the workflow management system, allocate more processors as needed. And this is the red line. And here we see that the utilization of the nodes follows pretty well the resources that have been allocated to it. If you're interested in BioBB, there are many example demonstration workflows on the website. I recommend you have a look at them. And the BioBB team is at the Institute for Research in Biomedicine in Barcelona, and also in the Barcelona Supercomputing Center. To finish with the presentation, BioExcel, in addition to development of all of these applications, we also have we are providing a lot of support services. We have support forums at ask.bioexcel.eu, where you can find support from the core developers and also from the community. We run very popular webinar series for many years now. There's 70-80 webinars at the moment. Monthly or bi-monthly, we have presentations, documentation, extensive documentation with tutorials, best practice guides, and a knowledge center is also available at docs.bioexcel.eu, and we also offer extensive training program. One of our main goals also is to strengthen the community and to engage closer with groups around Europe, in particular, but also around the world. And we've started this year an ambassador program where we have representatives in different countries. We also have in Greece, Zoe Cornea is our ambassador there, and we are planning to organize topical workshops with content. The content of those workshops will be based on the needs of the local user community. So we'll do a survey and see what exactly you need, and we will tailor that. So you can visit us at bioexcel.eu /ambassadors. You will find more about the ambassador program there and the planning for upcoming events. So that's all from me. I encourage you, if you're a user of any of the applications or the methods, to subscribe to our newsletter. It's a monthly newsletter, and to keep up with upcoming training events, for example, or other activities. Yeah, that's all from me. Thank you very much, and I hope you will find use of the applications and the services developed by BioExcel. Thank you.